It has now been established that modern humans have originated in Africa about 300,000 years ago. This is known as the out of Africa hypothesis. Previously, there was a competing theory that Homo erectus left Africa and settled in various continents around the world, where divergent subspecies or species evolved, and through a period of interbreeding that led to the rise of Homo sapiens. This was known as the multi-regional or multilineal model. Thanks to new discoveries in the fossil record and DNA analysis and research, the multi-regional model fell out of fashion in favor of the out of Africa hypothesis. Eventually, early Homo sapiens left Africa and migrated throughout the planet, settling in different ecological niches. The arrival of humans in North and South America is still debated, as new archaeological evidence is being discovered that potentially pushes back the earliest arrivals of humans in the New World from both Asia and potentially Europe and Africa directly. Often people ask, how do we date the fossils we find? There are several different dating methods. Specifically, there are four primary dating technologies for dating early hominids: radiogenic isotopes, paleomagnetism, thermoluminescence, and electron spin resonance. For early hominids like Australopithecines, radiocarbon dating would not be effective in dating those fossils. In the case of early hominid fossils, we use different methods like fission track and potassium orgone dating. Radiocarbon dating is effective for fossils and artifacts that are no older than 30 to 50,000 years old. Radiocarbon dating is used a lot in archaeology, and we will cover this dating method in more detail later. However, let us look at two other methods commonly used to date fossils: fission track and potassium orgone dating. Fission track dating is a radiometric dating technique based on analysis of the damage trails or tracks left by fission fragments in certain uranium-bearing minerals and glasses. Whereas potassium orgone dating is a method of dating rocks from the relative proportions of radioactive potassium 40 and its decay product, argon 40. The rate at which potassium 40 decays into argon 40 isotopes over time is known. Therefore, the ratio of potassium to argon 40 isotopes found in materials at different stratigraphic layers will give the relative age for that layer. This method is an example of radiogenic isotope dating method. The genetic record has also helped us to confirm the dates that correspond to our evolutionary history. We are able to compare ancient DNA found in fossils, if available, with modern DNA, and together we have been able to confirm our observations about our evolutionary processes and how we are related to other primates. Further, it has been learned modern humans have a limited genetic diversity indicative of population bottlenecking. This occurs when there is a drastic temporary reduction in the total population, thereby reducing genetic variability. Therefore, this would indicate, at some point in Homo sapiens development, an event occurred that drastically reduced the overall total population of humans, potentially only to about 2 or 3,000 individuals. Further, DNA analysis has demonstrated that the majority or greatest genetic diversity for Homo sapiens can be found in Africa. That means the majority of the variation in our DNA that we do have can be found within the continent of Africa. Evidence from mitochondrial DNA and the Y chromosome confirms this and confirms an African origin for our species. This is a basic evolutionary tree of hominids. It shows where we are within our history of evolution and how we are related to earlier hominids. We will now look briefly at some of these earlier species. However, included in this module are videos about some of these species for your review. I would like for you to watch these videos. Two videos, Lucy in Search of Human Origins Part 1 and, and the video Earliest Ancestors are long videos, and these are optional. I'm not requiring you to watch these two videos for the class. However, these videos are classic videos with regard to our evolutionary history. The Lucy video is a Nova special and, and part one of a three-part series of mapping out our human evolutionary history. I do provide links for the other two parts. I would like for you to watch the shorter videos. I will indicate which ones in the module.